If you listen to the critics, if you listen to your own doubts and call them reasons, you may never find the drive to go back to school. But a little change in direction, a new perspective, and the right program may make your successful new path a reality. The Accelerated Degree Program at Albright College. Start your degree or complete your degree one night a week in as little as 2 years. Albright College. A different way of thinking.
Good evening, and welcome to the Albright Scholar for July 2018. My name is John Pankratz. I teach history at Albright, and each month it's my privilege to welcome BCTV viewers and residents of Greater Reading to take a glimpse inside our learning community up here on North 13th Street, to look at the work being done by teachers and students, and to think a bit about its impact on all of us. Well, we have had just a couple glorious summer days recently. Just the sort of day that you'd want to, spend on the tennis court, or perhaps in the cell biology lab. Well, we don't have to choose today because my guest is Mara Trefoil, and she's a star in both venues. She is one of the dozen or so Albright students who are performing ACRE research, Albright Creative Research Experience. And she's also a star tennis player, being named the Player of the Year in the Mid-Atlantic Conference for 2 years in a row. Mara, welcome to the Albright Scholar.
Thank you very much, Dr. Pankratz.
Great. And we should establish that you're both doing your ACRE, but you're getting to play some tennis this summer as well.
Exactly. Good,
keeping all the muscles limber. What induced you to do an ACRE in the first place? It's a lot of work. And I think a lot of people look at the summer as a time to catch up or to make some money or to travel.
Yeah, exactly. I think it was my introduction to biology courses at Albright that really sort of pushed me to sort of discover research and kind of see what it was all about. Mm-hmm. And actually, several of the members in my service fraternity were working with Dr. Ventresca.
Right.
So they kind of pushed me to sort of talk to her and see what her research was all about. And when I went to talk to her and found out that she was working with adult stem cells and research regarding that, that was really interesting to me. And the ACRE program is really unique because it offers a lot of opportunities sort of outside research that you can get involved in as well.
No, it's a really special program. And I was glad to hear the genesis of your program, that it wasn't as if the teacher said, okay, I need an assistant to help me with this. It's your own interest, your own curiosity, and then you approached the teacher and discovered an area of common interest and a project that she could very much foster with you. Yeah. Now, had you had Dr. Ventresca in class before?
So actually I had not,
okay.
But while I was TAing, so as a teaching assistant for some of the introduction to biology courses, mm-hmm. Alice Brylowski suggested that I go talk to her and Dr. Mack and kind of see what research I might be interested in and where to sort of take my path with that.
Okay, good. And it's working out well.
Yeah, I've really enjoyed it so far.
Well, I'm going to ask you to pronounce all the PI3K names, but give us just the elevator glimpse of your research project, and then we'll delve into some of the procedures you're going through.
Yeah, sure. So essentially, we sort of wanted to take a look at how diet, and because diet has become such a prevalent problem in mm-hmm. today's society, mm-hmm. and how that can affect adult stem cells. So essentially what we're doing is we're feeding fruit flies, scientifically known as Drosophila melanogaster. Mm-hmm. We're feeding them increased levels of sugar and artificial sweeteners and seeing how that affects their adult stem cells. And the way that we're doing that is by using egg laying as a model for that. So essentially we see that if their egg laying becomes, like if they lay a lot of eggs, then that means that their adult stem cells are not doing as well, or doing well. But if mm-hmm. they lay less eggs, that means that their adult stem cells are not doing well. So essentially we're using that as a model to gauge how well their adult stem cells are doing in their bodies.
Right, and sort of keeping track of how they're aging as well?
Yes. Mm-hmm. Mm-hmm. So essentially adult stem cell health is often related to these different diseases. So when your adult stem cell health has become compromised or is mm-hmm. not going as well, that often leads to the development of various diseases, such as cardiovascular disease, cancer, obesity.
This classic diseases of old
exactly.
age.
Mm-hmm.
And it's down at that cellular level that your body's telling you you're getting old.
Mm-hmm.
Yeah. And maybe there are ways to adjust that if you understand the mechanism. So you're not doing sort of microbiology here. You're looking not with a 10,000 power microscope, but maybe a 300 power microscope?
Yeah. So really, the only time we kind of use the microscope in the lab is to see how many eggs that were laid. So because the eggs are about 10 millimeters in length, so it's really kind of hard to see how many they laid. And also to differentiate between males and females in the lab, so we can know how many to put in each chamber.
Right, so that they can meet each other.
Yeah.
Yeah, yeah. And how do you do that? How do you look at a Drosophila and say, oh, that's a boy, that's a girl?
So the males tend to be smaller in size, and they have a dark spot kind of at the bottom of their abdomen.
Okay.
And the females tend to be larger and lack the dark spot. There's a few other things, but it gets a little complicated.
Right, right. And how do you get these individuals?
So we kind of house them in the lab, and they're usually incubated at about 25 degrees Celsius. And essentially, for our research, we always want like young, mature flies, so that we know, because just as with humans, how aging kind of slows down your cell division, the same happens with the flies.
I bet.
So we essentially will remove flies from a vial and allow new ones to hatch. So then we know they're kind of 1 day old, and when they're 5 days old, they're mature to us, so we can go ahead and start using them.
Great. And as we know, over the years, lots of research has been done using this fly as a subject, right?
Yeah.
Why is that?
Well, they're really a great organism to use in the lab because they have short life cycles. They're really easy to replicate. Like, we have so many vials, and they're just so easy to keep track of. But also that they have very similar genetic counterparts to humans. So that allows us to kind of compare in some manner what happens in the flies to what could happen potentially in humans.
Right, and so you can study that mechanism.
Exactly.
A lot of genetic research has been done on fruit flies, but then you're talking about these various sort of signaling devices between receptors and certain enzymes, and then certain products for the adult stem cell. Yeah, and nobody cares that much about fruit flies, right?
Yeah, unfortunately, not so much. Yeah.
So nobody's demonstrating outside the lab.
Yeah.
I don't think.
Yeah.
I don't think. And is this research that Erin herself has conducted in the past, or a related study?
So she's worked with other students in the past, not specifically on this study
Right,
with
I
this
think it's
high
been different,
sugar increase and artificial sweetener increase in the fly diet. But they have manipulated different parts of the flies and different things with their diets, but never kind of this sugar diet.
Now you say you're using both sugar and artificial sweetener. Are you using those separately, or are you just loading all sorts of sweet into the fruit fly diet?
So it's separate. We wanted to compare exactly how, so the research kind of has two parts. The first looking at how many eggs do the flies lay as a result of the diet they're being fed. And the second part has to do with how long these adult stem cells are able to survive in their egg-laying structures. So with that being said, we're kind of doing, sorry, what was the question again?
Right, whether you're doing a separate group of flies on artificial and a separate group on sugar.
So with the artificial sweeteners, we're feeding them the increased level of artificial sweetener and then comparing it to how the increased sugar works. So essentially each vial and each chamber has, like for example, chamber A has increased level of sugar, chamber B has an increased level of artificial sweetener, and so forth. So we're kind of able to compare how the sugar affects the flies versus to how the artificial sweetener affects the flies.
Right. Now, is there anything nutritional in your vials as well?
Yes. So
so there's actual food besides all this.
So the flies. That's a good point. So the flies usually use yeast as a carbohydrate source, or yeast as a protein source, and sucrose as the carbohydrate source. So we always will mix in yeast as well, and typically a cornmeal, and it just kind of depends from the first part to the second part of the experiment.
Okay. Do you have any guesses what you're going to find out?
Yeah, so we kind of expected that for the first part of the experiment where we're looking at these egg-laying totals, we expected that the sugar would allow the flies to lay a lot of eggs initially.
Yeah.
However, for the second part where we're seeing how long they're able to lay these these eggs, so how long is it before their stem cells become exhausted as a result of their increased sucrose diets. So with that, we expected increased levels of egg laying initially. However, we expected that the increased sugar would not allow the flies to lay eggs for a long period of
Right,
time.
their procreative lives would be shortened.
Yeah, and with the artificial sweeteners, we kind of expected them to be able to lay flies for a longer period of time because they're not really activating the same pathway.
But they might
with the
have these
sugar.
terrible migraines as well. And
Yeah.
I shouldn't joke about that. It's sort of true. Which artificial sweetener are you using?
So we tested a bunch initially
you
to
did,
see
okay.
which ones actually had an effect greater than the control, which is just the regular yeast. And the ones that we're moving forward with are the Splenda and the Equal sweeteners. Yeah, so those are the ones that kind of increase
So
the
both
egg
sucralose
laying.
and aspartame.
Yes.
Okay.
There you go.
Good. I wasn't going to name the brands there just in case they were upset by this. Oh, well, this is absolutely fascinating. So, and because this whole cycle that the flies are going through, that can happen over a summer, right?
Yeah. So for about the first part of the summer, we worked on the egg laying part of the experiment. And right now, we kind of started to transition to the survival part of the experiment. So the egg laying was kind of to see which of the artificial sweeteners actually had an effect on the flies greater than the control. Because if they didn't have an effect greater than the control, the sweeteners could just be toxic to the flies. We didn't quite know what the effect was.
What was the mechanism that was affecting that
Yeah.
result? Okay. And so you've got all that groundwork.
So now
laid.
we're moving in to see how long do these adult stem cells survive in the flies as a result of their increased sugar or artificial sweetener intake.
How fabulous.
Yeah.
Yeah. Well, this ought to come up with something really fascinating. Are you and Dr. Ventresca hoping to publish the results?
Yeah, hopefully. We kind of wanted to see sort of what would happen over the summer acre, and we actually talked maybe a little bit about continuing the project and sort of replicating it further.
Right. Certainly a number of students have taken the boost given them by the acre and then translated that into a senior honors thesis and everything. You're only a junior now, so you have some time to decide on that kind of project. I know you have pre-medical interests. Do you see a connection between this research and what you'd be learning or pursuing in medical school as well?
Yeah, definitely. I think diet and how we sort of manage our diet has become such a prevalent topic in today's society that I think just kind of being able to sort of study this on a smaller scale with the fruit flies and see, sort of what this increased level of sucrose or artificial or sweetener can do to an organism, I think can really sort of help me understand, what kind of preventative measures people can take. And as a physician, I would be able to sort of steer people in the right direction in terms of their diet.
Right. While talking to them about their intracellular mechanisms and everything like that. We don't necessarily think of it that way, but that's the way nutrition works. That's the way our bodies work. It's not simply the big things we can see, but it's these tiny little sort of chemical reactions within our cells that determine their health and our overall health as well. Wow. What would you say to, well, you said that you were advised by some of your fellow service fraternity members to explore the possibility of an acre. What would you say to, say in the fall you meet a talented freshman science major, what would you say to them?
I would highly recommend it. I think it's an awesome opportunity to kind of explore research for the first time or, for a continued experience and sort of do it in such a warm and welcoming environment. Like we have these summer luncheons where we get together and discuss our different projects, and it just feels like such a great close-knit community, and it's been such a great experience to kind of talk about my project along with others and sort of, figure out better ways to go about different things, and it's been such a collaborative experience.
And all the Acre students are living out in the Albright Woods apartments.
Yeah, so it's been a really fun
little community there.
experience, yeah.
As well. You mentioned these Acre lunches, and I'm a great aficionado of those. I love coming because it exposes me to areas outside my expertise. Have you enjoyed hearing the psychologists and the communication students and the religious studies students
Absolutely.
talk about their work?
I think just how they combine their different interests. I know one of the students is combining her interest in computer science and video games with her interest in humanities and history and archives, and how she was able to sort of combine those two interests into a research project is just fascinating. And there's many more like that, so it's
Right.
really exciting.
In some ways, it's an outgrowth of Albright's nature as a liberal arts college. Right?
absolutely.
Because while you're a biology major, you're also taking courses in the humanities, in the social sciences, in the arts.
Yeah.
And careful thinking, imaginative thinking, I think comes from everything that human beings do. And so it's not as if there's a direct path from this course to this major to this profession. Everything feeds and fertilizes that pursuit. Yeah. So med school, grad school, have you begun to narrow that down yet?
Yeah, I'm interested in going to medical school, and I've been able to participate in a lot of different opportunities that Albright offers. Like one of them is the Emergency Department Scribe program
Right.
in the spring, so I was able to do that, and I was able to work with a physician and sort of shadow him, but also take on a role as a scribe in the emergency department. So that was very eye-opening.
Literally taking the notes
Yeah,
and
exactly.
keeping track of the patient diagnosis and treatment and that sort of thing. Is that a harrowing experience as well?
Yeah, it was really a wonderful experience, just kind of getting exposed to that department of medicine, and I built such a close relationship with the physician, so that was really unique as well.
And that means so much. And I think very often physicians, because they've been through the process and because they've had junior colleagues, can spot that kind of talent and that temperament, right? Not just the smarts, but the ability to think under pressure and to come up with solutions and everything like that. So yeah, and that's important. I mean, medical school costs a lot of money, and so you don't want to go into it without a sort of mature and experienced set of motivations. Boy, yeah. Well, that will be great.
Yeah.
That will be great. Now, have you given your presentation yet? I was away on a trip, which is not like me, but I did it. And so I missed a couple of the lunches.
Yeah, I did. I think it was last week, actually.
Okay.
I believe so. Or no, this past, well,
The week before,
yes,
yeah, because
this was the 4th.
it was the 4th. And how did that go?
I think it went really well. A lot of the people that were in the room sort of were very friendly about it, and said that they really understood kind of what seemed to be a complicated topic at first. So that was good to hear.
Yeah, and that's the other challenge. Not only do you have to understand what the artists and the social scientists and the computer scientists are doing, but you have to make yourself understandable to those people.
Yeah.
And that's a great challenge. Is bringing highly complex forms of analysis down to a level, or translating it into terms that the layperson can understand. And I think that's such an important thing. Right? I don't know whether people enjoy higher education or respect higher education the way that they should. And so I think the more adept we are at talking about how we think and what our procedures are, the more I think understanding and appreciation will be out there. So how many hours a week have you been spending in the lab?
Several, yeah. Probably about 4 hours a day or so.
4 hours a day.
Yeah. Depends. Each day is a little different. So some days take longer when I have to prepare the food for the flies. Other days are a little quicker when it's just moving them to a new vial. But it just kind of varies. And then some of the other time has been spent just reading articles and sort of preparing for presentations and elevator pitches and different things of that nature.
There's a great deal of scientific literature around all of these mechanisms. And it's sort of a cutting edge area of research. It's something that lots and lots of scientists are trying to make sense of because of the potential therapeutic effects as well. I guess what you're talking about is not only aging, but those various diseases that come with old age, diabetes and heart disease and cancer.
Yeah, exactly. And those are all linked to sort of this loss in adult stem cell health, which, as we're kind of reading in our literature and sort of putting into our project, is often related to what we eat, and how diet can sort of transform metabolism to act in such a way that's harmful to the body.
Right. So I've been invited out for ice cream cones on Wednesday. Should I probably go out and have a salad instead, shouldn't I?
That would probably be better, yes.
Or maybe if I took up tennis and played a lot of that.
Yeah,
Yeah.
that would be a good option as well.
That would be the other way to metabolize all those
Sure.
excess inputs and that sort of thing. Great. Well, it sounds like it's just been a great summer.
It really has been.
I'm sorry that Erin couldn't be with us tonight. She was unavailable tonight. But she's been on the show before, and I know that she's worked with students on ACRES before, and that mentoring relationship is just a great one.
Yeah, she's been really great and really helpful.
And do you have courses with her coming up then in the,
Yes,
in the next 2 years?
but I plan on taking several of her classes, so I'm excited about that.
And there are other faculty members who are interested in this level of research, right? The cellular level. I know Dr. Hirschberger is as well. He must have good questions for you
Yes,
as well as commentary. Good. So it's in general just a really encouraging kind of environment in which to do science or to do research of any kind.
Yeah, it really is.
Good. Well, thank you so much for being with us today, Mara.
Thank you so much for having me.
I'll let you go now, and we'll welcome our viewers to tune in another month. We'll be talking about more ACRE projects. I do have several people who have already committed to an August appearance, and we'll look forward to learning more about what people are doing to make the summer a really stimulating and profitable time on the next edition of the Albright Scholar. Good night.
If you listen to the critics, if you listen to your own doubts and call them reasons, you may never find the drive to go back to school. But a little change in direction, a new perspective, and the right program may make your successful new path a reality. The Accelerated Degree Program at Albright College. Start your degree or complete your degree one night a week in as little as 2 years. Albright College, a different way of thinking.